Insights, Basics and Beyond. Chapter 8, Natural Killer Cells. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. Last time, we explored the fascinating world of dendritic cells, the immune system's sentinels that play a crucial role in bridging the innate and adaptive immune responses. Today, we are diving into another key player in our immune defense, natural killer cells or NK cells. These cells are part of the innate immune system and serve the body as first defense line against viral infections and tumor formation. Natural killer cells are unique in their ability to recognize and eliminate cells that are stressed, infected, or transformed into cancer cells without the need for prior sensitization. This makes them incredibly efficient and effective in responding to threats that require immediate action. So let's talk science. What are natural killer cells? Well, NK cells are a type of lymphocyte, a white blood cell that circulates in the blood and lymphatic system. They originate in the bone marrow and belong to the innate immune system. Unlike T cells and B cells, which rely on specific antigen recognition, NK cells have a more generalized role, enabling them to act swiftly when encountering abnormal cells. NK cells are named after their ability to naturally kill cells that are harmful to the body. They excel at recognizing changes on the cell's surface, such as the absence of major histocompatibility complex class I molecules or MHC1 molecules, or the presence of stress-induced ligands that signal infection or malignancy. This ability to identify and respond to abnormal cells without prior exposure makes NK cells indispensable in early immune responses. NK cells constitute a bridge between innate and adaptive immunity. NK cells, though part of the innate immune system, display unique features that blur the boundaries between innate and adaptive immunity. Unlike most innate immune cells, subsets of NK cells exhibit a form of immunological memory, enabling them to mount faster and stronger responses to repeated infections, such as those caused by cytomegalovirus or CMV. This memory-like behavior, driven by activating receptors and epigenetic modifications, represents an evolutionary link between the immune system's two branches. A key example of this link is the striking similarity between NK cells and cytotoxic T cells or CD8-positive T cells. Both cell types utilize perforin and granzymes to kill abnormal cells, though NK cells act broadly and without prior sensitization, while T cells require specific antigen recognition. These overlapping mechanisms highlight how the immune system layers innate rapid responses with adaptive specificity for optimal defense. Beyond their functional parallels, NK and T cells also collaborate dynamically. NK cells secrete interferon gamma or IFN gamma, which primes T cells for enhanced activity, while T cells release interleukin-2 or IL-2, amplifying the NK cells' responses. This cytokine-driven interplay exemplifies how the innate and adaptive arms of immunity regulate and bolster each other, creating a robust and coordinated response to viral and other infections as well as cancer. NK cells therefore serve as a versatile bridge, uniting rapid innate action with the precision of adaptive immunity. Let's have a look at the mechanism of how NK cells act. NK cells utilize two key mechanisms to detect and destroy their targets. First, missing self-recognition. Normal cells express MHC class I molecules on their surface, which act as a please do not kill signal for NK cells. Cells that lose these molecules, often due to viral infection or transformation into cancer cells, trigger NK cells to attack. Second, activating receptors. NK cells also have receptors that recognize stress-induced ligands on abnormal cells. When these activating signals outweigh inhibitory signals, the NK cells release cytotoxic granules containing perforin and granzymes. Perforin forms pores in the target cell membrane, allowing granzymes to enter and induce apoptosis, which is the programmed cell death. This dual mechanism ensures that NK cells can quickly eliminate dangerous cells while sparing healthy ones. NK cells need to balance inhibitory and activating signals. The activity of NK cells is tightly regulated by a balance between inhibitory and activating signals. Inhibitory receptors recognize normal MHC class I molecules on healthy cells, sending a do not kill signal. Activating receptors detect stress signals or abnormal molecules on the surface of potential target cells, initiating cytotoxic action when dominant. This intricate balancing act prevents inappropriate NK cell activity, ensuring that healthy cells remain unharmed while infected or cancerous cells are destroyed. NK cells play a critical role in immune surveillance and cancer, continuously monitoring the body for signs of infection or malignancy. In cancer, NK cells are particularly important in identifying and eliminating tumor cells that downregulate MHC class I molecules or express abnormal levels of stress ligands. However, tumors can evade NK cell surveillance by creating an immunosuppressive environment or by upregulating inhibitory molecules. This has fueled significant interest in developing therapies to enhance NK cell activity, especially in cancer immunotherapy. NK cells are also crucial in fighting viral infections. Many viruses evade immune detection by downregulating MHC class I molecules to escape cytotoxic T cells. NK cells counter this strategy by targeting infected cells that lack these molecules. This ability makes NK cells particularly effective against viruses like herpes viruses and cytomegalovirus, where a rapid immune response is essential to contain the infection. While NK cells are vital for immune defense, their dysfunction can contribute also to various diseases. NK cell deficiency, a rare genetic or acquired condition, can impair NK cell function, leading to increased susceptibility to viral infections, especially herpes viruses, and cancer. Excessive NK cell activity, on the other hand, can cause immune disorders such as hemophagocytic lymphohistiocytosis or HLH, where excessive inflammation damages tissues. Last but not least, impaired NK cells may allow tumors to evade immune surveillance and thereby increase the risk for cancer. These conditions highlight the delicate balance required for NK cells to function effectively without causing harm. However, even if I just implicated NK cells in diseases, they also have a tremendous therapeutic potential. Their ability to recognize and eliminate abnormal cells without prior sensitization makes them ideal candidates for immunotherapy. First, we have adoptive NK cell transfer. NK cells can be isolated, expanded, and activated in the lab before being reintroduced into the patient to enhance immune responses against tumors or infections. Second, we have antibody-based NK cell-mediated therapies. These antibodies can enhance NK cell targeting by bridging them to the target cancer cells. Last but not least, CAR-NK therapy. Similar to CAR T-cell therapy, which we will cover in a later chapter of this podcast, genetically modified NK cells can be programmed to recognize specific cancer antigens by transgenic CARs. These approaches hold promise for treating cancers, chronic viral infections, and even autoimmune diseases, offering hope for patients resistant to conventional therapies. To conclude, natural killer cells are a vital component of the immune system, acting as the body's first line of defense against viral infections and others, as well as cancer. Their unique ability to rapidly recognize and eliminate abnormal cells underscores their importance in maintaining health. As we continue to unravel the complexities of NK cells, their therapeutic potential becomes increasingly apparent. From cancer immunotherapy to antiviral treatments, harnessing the power of NK cells could revolutionize immune-based therapies. In our next chapter, we will delve into B cells and the process of antibody production, a cornerstone of the adaptive immune response. B cells are the architects of immune memory, enabling the body to respond more effectively to pathogens it has encountered before. Join us as we explore the fascinating world of antibodies and long-term immunity. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.